Okay, let's just dive right in. We're witnessing, I think, one of the most exciting transformations in molecular science right now.
Mm.
And it's not just about making things a little bit better. We're seeing AI completely shatter these decades-old ceilings, both computational and experimental.
Oh, absolutely. It's a huge
We're
shift.
moving beyond simple prediction into, true generation and just radical acceleration.
That's the perfect way to put it. For so long, researchers have been stuck on these three massive bottlenecks,
Right.
simulating how proteins move, designing brand new molecules from zero, and just getting a computer to actually understand the rules of chemistry.
And the sources we've pulled for today's Deep Dive 3, really cutting-edge reports,
Mm.
they show AI crossing all three of those barriers basically at the same time.
Exactly. So first, we're looking at DeepJump. It uses AI to speed up those incredibly slow protein movement simulations, molecular dynamics, or MD.
Okay.
Second is BoltzGen. This is a big one. It's a universal generative model for designing things like therapeutic proteins and nanobodies for drug discovery.
And the third.
And finally, ChemIQ. It's a new benchmark designed to test if these large language models, LLMs, can go beyond just matching patterns and actually reason about organic chemistry.
So our mission today is to really get into the mechanics. How do these things actually work?
Right.
What are their, real-world success rates in the lab? And what does it all mean when you put them together?
Let's do it.
So, Deep Jump. Let's start there, because it tackles this classic, just brutal simulation challenge. If you're trying to watch a protein fold, classical MD forces you to use time steps measured in femtoseconds, a quadrillionth of a second. Why? Why so microscopically precise?
It's all about the physics. You have to capture the highest frequency atomic motions, things like bond vibrations.
And it wiggles.
Exactly. And if you skip those, if your time step is too big, you violate the underlying laws, and your whole simulation, it just becomes inaccurate almost instantly.
So simulating something that happens in, say, a microsecond in real life.
Would take centuries of compute time. It's been this impossible wall for decades.
So how does Deep Jump get over that wall? The paper calls it a generative model utilizing Euclidean equivariant flow matching. Can you break that down for us?
Yeah, let's unpack that. So flow matching is technical, but you can think of it like this. Instead of calculating where every single atom goes every single femtosecond.
The step-by-step march.
Right. DeepJump is trained on huge data sets of protein snapshots. It learns the generalized pathways of motion.
I see.
And that Euclidean equivariant part is so important. It just means the model intrinsically knows that the physics of a molecule don't change if you just, rotate it in space.
So it has some built-in physical intuition.
It does, and that allows it to predict how a protein will change its shape across much, much bigger timescales.
So it's taking these massive temporal jumps. It's not marching, it's leaping. It says, okay, I predict that in, say, 10 nanoseconds, the protein's going to look like this.
That's it exactly. They call that jump size delta, or delta in the paper, but yes, and the results are just, they're stunning.
What kind of results are we talking about?
They demonstrated roughly 1,000-fold computational acceleration over traditional methods.
1,000 times faster.
1,000 times. That's the kind of leap you need to make simulating complex proteins a routine thing, not a massive supercomputer project.
Okay, 1,000-fold is revolutionary.
Yeah.
But was it just faster, or was it also accurate? Did they, gut check the physics?
They did. They validated it using what's called ab initio folding simulations.
Which means starting from scratch.
Exactly. You start with just an unfolded string of amino acids, and you ask the model, okay, fold this into its final correct shape.
It's a real test.
The real test. And for well-known fast-building proteins like WW domain and NTL9, DeepJump successfully reproduced the entire folding pathway. It found the correct native states.
Wow. So it generalized beyond what it was trained on.
It did. But here's where it gets really interesting, because there's a trade-off. You can't just crank that temporal jump size, that delta value, to maximum and expect perfect results, right?
I was going to ask about that. There has to be a catch.
There is. And the researchers did a beautiful job mapping it out. If you choose a really big jump size, like 100 nanosecond steps, you get the absolute fastest simulation.
Yeah.
But the quality degrades a lot. They found those simulations sometimes failed to fold the proteins at all.
So speed kills accuracy if you're not careful. You literally jump over the most important moments.
Precisely. Protein folding is all about these rare barrier-crossing events. A protein can sit in a stable state for, 99% of the time. And then in a fraction of a nanosecond, it crosses an energy barrier to get to the next step. If your jump is too big, you just leap right over that crucial moment. The model gets lost.
So what's the sweet spot?
They found that smaller steps, 1 nanosecond, maybe 10, were necessary to really capture those fine-grained kinetics and get to the right final structure.
So the big takeaway here is that breaking the timescale barrier is huge, but true utility is about balancing that speed with the precision you need to see how biology actually works.
Right.
Okay, so now that we can simulate motion faster, let's pivot. Let's go from watching things move to actually creating new things, new functions.
And that takes us straight to BoltzGen. We're moving from analysis to design.
BoltzGen is described as a single all-atom diffusion model. I mean, that sounds a bit like the holy grail of molecular engineering. Why do they call it universal?
That universality is the key. Most of the generative models before this were really specialized. You have one for small molecules, another for proteins.
Right.
BoltzGen is a single model that can generate DNA, RNA, small molecules, proteins, you name it.
Yeah.
And it's an all-atom model.
Meaning it's building the whole thing at once.
The whole thing, the 3D structure, the XYZ coordinates of every atom, and the sequence of amino acids all at the same time.
That sounds unbelievably complex. If I'm a researcher, how do I tell this universal model what I want it to design?
That's where its rich design specification language comes into play. It gives you incredible control. You don't just say, make me a binder.
You can be more specific.
Way more specific. You can say, I want a protein with a helix right here. Or, I need this specific residue to form a covalent bond with my target. You can even tell it which parts of the protein should be doing the binding and which parts shouldn't.
That's a level of control we haven't really seen before. But the real proof is always the wet lab validation, right? Especially against tough new targets. Did they really challenge the model?
They did. They deliberately picked difficult targets, things with a structure that had very little similarity, less than 30% sequence identity to anything the model had ever seen in its training data.
So it's a true test of generalization, not just memorization.
Exactly.
And what happened? What were the results against those 9 novel targets?
The results were, frankly, really encouraging. BoltzGen successfully designed high-affinity, NM, that's nanomolar binders, against 2/3 of them.
66%?
Yep, 66%, across both nanobody designs and more traditional protein designs. And nanomolar affinity is well within the range where you start thinking about actual therapeutic potential.
Can you give us a tangible example? What could these designed proteins do?
Sure. They were able to design proteins that could target and neutralize these highly bioactive, sometimes toxic peptides.
Okay.
For example, they made nanomolar binders against melittin and indolicidin. These are peptides that can destroy red blood cells or kill bacteria. The designed proteins bound to them so tightly, they basically just turned off their dangerous function.
Wow. But, like DeepJump, BoltzGen has its own limitation, its own trade-off. They found what they called the ubiquitin memorization issue. What does that tell us?
It's a classic machine learning failure mode, and it's brilliant that they included it in the paper.
Can happen.
When they asked the model to design proteins in a very specific narrow size range around 73 to 76 amino acids, the model's creativity just collapsed.
It stopped making new things.
Completely. It just started spitting out sequences that were over 97% identical to the structure of ubiquitin.
And why ubiquitin of all things?
Because ubiquitin is this fundamental biological tag, and it is massively overrepresented in the training data, the PDB. It's in thousands and thousands of structures.
So the model learned that for a protein of that specific size, the safest, most probable answer was just ubiquitin.
Exactly. The sheer volume of that one example overwhelmed its ability to generalize in that narrow window. It's a great reminder that these tools are still shaped by the data you feed them.
But the team isn't hiding that. They're being transparent.
Absolutely. The whole ethos behind BoltzGen is about democratization. The model is totally free, open source. The idea is to get these state-of-the-art design tools into the hands of any researcher, anywhere.
That's fantastic. Okay, so we've covered acceleration and simulation with DeepJump and creation and design with BoltzGen. Let's shift gears to intelligence, abstract intelligence. Can a language model actually reason about a molecule, or is it just a really sophisticated parrot, spitting out text that looks like chemistry? That's the question ChemIQ is trying to answer.
And it's such a deep, important question. ChemIQ was designed specifically to avoid tests you could easily game.
No multiple choice.
No multiple choice. It has 816 questions that require short, specific answers. They're testing whether the model can build an internal representation of a molecule and apply chemical theory to it.
And the results between models designed for reasoning versus those that weren't. It was night and day.
It was an immediate separator. The standard, non-reasoning LLMs were terrible. They scored between 3% and 7% accuracy.
Basically guessing.
Basically. But the models trained for explicit reasoning, O3 Mini, Gemini Pro 2.5, DeepSeek-R1, they scored between 50% and 57%.
That's a huge leap.
It confirms that the ability to, show your work, to articulate those intermediate steps, is what unlocks real problem solving in an LLM.
So let's look at an example. They tested it on finding the shortest bond path between two atoms in a chemical notation called a smile string.
Right, which sounds simple, but it requires abstract thought. A human chemist sees that string and mentally builds a graph of the molecule in their head. Atoms or nodes, bonds or edges.
And then they search that graph.
Exactly. And the results show these reasoning models can do that too. The O3 mini model answered 89% of these questions correctly. That strongly suggests it's building and navigating a real molecular graph internally.
What about going from that smiles notation to a formal chemical name, the IUPAC name? That's always been a disaster for language models.
A total disaster. It was basically a 0% task before. Now we're seeing real success. Gemini Pro 2.5 got between 35% and 44% accuracy.
Which is significant.
It's very significant because it proves the model can map this complex, structured notation to established chemical vocabulary. It's understanding the rules, not just the letters.
But the absolute gold standard they tested was NMR structure elucidation. This is where a chemist gets multiple pieces of complex data spectra, a formula, and has to put it all together to figure out the molecule structure.
It's pure synthesis and deduction. And Gemini Pro 2.5 was the star here. For simple molecules, it was solving the structures with 73 to 94% accuracy.
Incredible.
But the real headline was its ability to handle complexity. It successfully figured out the structure of a complex molecule with 25 heavy atoms using 2D NMR data.
Wow.
That kind of multimodal data integration and deduction. I mean, it really suggests these LLMs have reached a level of abstract chemical understanding that, until now, was pretty much limited to human experts.
So we've covered this incredible trifecta in molecular science. With DeepJump, AI is driving acceleration, making simulation 1,000 times faster. With BoltGen, AI is driving creation, designing high-affinity binders for new targets. And with ChemIQ, AI is showing genuine intelligence, reasoning about chemistry at an expert level.
And they're all completely interdependent. We now have the tools to rapidly model systems, design new parts for them, and the intelligence to interpret the chemical rules that govern it all.
So the bottleneck shifts.
The bottleneck shifts right back to the physical world. How fast can your wet lab actually synthesize and test the flood of high-quality designs these AIs are now able to generate?
That's the perfect way to frame where we are now. But I want to leave you, our listener, with one last thought. It goes back to DeepJump.
Okay.
We established that to get the highest fidelity, the most accurate simulation, you needed those small, precise time steps, that little delta, to capture those rare but critical barrier-crossing events in protein folding.
Right, the details matter.
The details mattered. So as models like BoltGen and these reasoning LLMs get even better at generalizing and abstract understanding, how long will it be until an AI can accurately simulate those rare events, achieving both massive acceleration and high fidelity without needing to train on any existing trajectory data? Instead, relying purely on fundamental learned physical laws.
Wow.
Think about that gap between learning from data and just knowing the physics.